Okay, so now I do the introduction. Welcome to this new cohort of the Master of Science in Bioethics at St. Thomas University here in Miami Gardens. Starting this fall of 2019 is already the fourth cohort of the program. And I always like to begin with a little prayer. In the name of the Father, the Son, and the Holy Spirit. Blessed Trinity, we love you, we adore you, and we thank you for all your many blessings, for the gift of life, faith, hope, and charity, for having us here today, Lord, to start this new cohort in the Masters in Bioethics. I pray for my new students that they may enjoy this program, that it may be challenging, and that we remain all open to what you want to work in us during this time that we are together, Lord, for the next 15 months, as we delve into the issues of bioethics that have to do with nature, the nature that you have created around us, including our human nature in your image and likeness, and the dignity and the sanctity that we receive from you to put it into practice so that we may be ethical leaders, as St. Thomas wants to promote ethical leaders for life, to have respect for human life, respect for the environment, especially very much at this critical time of our history here on Earth. Lord, we realize and we know that we are the only species capable of destroying the Earth, and yet you have put us here for stewardship, for care of creation. We thank you for this challenge, for this opportunity, and we take it seriously as we seek to integrate these issues of bioethics into our daily life. We pray for all teachers or students here at St. Thomas and throughout the world as we begin this new academic year in the fall season, that it may all be according to your divine will. In a special way, we pray for the people of the Bahamas, for all, also in the Carolinas, and all those in harm's way of human or natural disasters. A couple of hurricanes also in the Pacific Ocean, going toward Japan, going toward the Korean Peninsula, for all your people, especially those who are in most need of your divine mercy. This we ask in the name of Christ our Lord. Amen. Father, Son, Holy Spirit. Okay, forward. I'm anxious to get into the meat of it, which is this first course. And also remind me to say a few words of the other course. Might as well do it now. So we have this first semester, we have 513 and 514. They're all bio courses. Back up a little further. Actually, there are 8 courses in the program, 8 courses, and we do 2 courses per semester, okay? And so the university is doing one thing and I'm doing something else, but you'll see how it all fits together. That's how I managed to put 8 courses in these 4 semesters, which will be fall, spring, short summer, summer 1, and then we all get summer 2 free, hooray, for about 6 weeks, and then come back for the second fall, so that you will be graduating by December of next year. And in fact, the third cohort is now in the fall. This is their second fall, all right? And at some point I want to organize a little reception, cocktail, something, get together so that you meet at least some of the folks from the previous 3 cohorts with spouses. We'll make it a nice social get together, all right? Probably toward November, December. Okay, so 2 courses per semester. That means that fiscally it will be about $4,000 that you'll have to be dishing out toward the beginning of each semester. So 4 times 4 will be about $16,000 for the whole program. It's about about $560 per credit, more or less, which is the least that I could get it for the program, times the 8, times 30 credits, which is the minimum for a master's program. So I've kept the numbers to the bare minimum so that it's the least expensive for the student. But for that, you're going to get a lot of bang for that buck because I'm going to give you a lot of material to think about and to reflect, all right? So this is the way we work it. We have, within each semester, we have the full length, the 16-week semester, but we also have the option of 2 mini semesters of 8 weeks each, and I'm capitalizing on that to fit the 2 courses per semester that way. So for example, on the books, you will see that this course, the 513, is on what we call Fall 1, FL1, which is supposed to be the first 8 weeks. Pay no attention to the man behind the screen. We're going to go the whole 16 weeks, okay? That way, that's how I'm able to fit this course, because 8 weeks is not enough to cover the material that we're going to cover here. But then, what I do with the other one, the 514, which technically is scheduled for Fall 2, the second half, right? Forget about that too, because that's actually going to be online for you, and you'll be able to start on it right away, all right? Because I'll give you materials to do that. And that will also involve the summaries, the typical summaries that I talked about, okay? But that one's going to be basically online. And that 514 involves 2 things. It involves downloading this book that is called On Being a Scientist. And I'll send you this information by email, but basically On Being a Scientist is a little book that is put out by NAS, which is the National Association of Science, National Academy of Science of the US, all right? National Academy of Sciences, they put out this nice little book. It's free online, has about 20 chapters, but each chapter is about 2 pages. So I'm gonna ask for a summary of each chapter, all right? And you'll be working on this throughout the whole thing. You need to complete this by the end of November or beginning of December, so I can have a grade for you on this one. And that's one part of the course. The other part of the 514 course is 3 modules that you'll be doing online also. 3 like little certifications from an ethics core, an ethics center that is at the university, is housed at the University of Illinois, Urbana-Champaign. My brother used to teach there last century, and in social science, not this one. But anyway, they got a grant about 5 years ago, a $5 million grant from NSF to develop a national ethics center for responsible research and professional conduct, all right? So because it's an NSF grant, which is public money, it's for free online and we have access to it. And the idea is that they have these little modules that one does. You read some stuff and then you'll be asked some questions, you answer that, and when you get them all right, or like 90% of them right, then you get a little certification. And there's 3 levels. So you'll do that online. So you have those little certifications to do, plus a summary of this. And that's the 514 course, which is basically responsible research and professional ethics. We'll talk about, you know, not plagiarizing information and copyright, and we'll talk about intellectual property and things like that, how to handle data, proprietary information, and just how to be a responsible research in the lab or in the professionals like nursing and doctors and so forth. Okay, more about the 514 later on, and I'll send you that online, so look for my email on that. Also, because now the next lecture will be in 3 weeks. We'll look at the little calendar. It will be the last Saturday of September, okay? Because the next 2 Saturdays I have commitments that I couldn't get out of, and therefore we'll meet again the last Saturday of September. So you'll have some extra time. I'm gonna give you stuff to do during these weeks, including the first 3 chapters of Meyer, which is our textbook on loan for this semester, for the 513 course. Yeah, and that's the book that you have there for loan for the semester. You'll hear more about Meyer in a few minutes. All right, questions and comments. I'm a little bit all over the place, but just stop me if you have some questions. Otherwise, it's all recorded, so we can all go back to it and say, what did I say? I can go back to the recording and see what I said. All right, let's get into it. So these are fundamental principles in Catholic bioethics. My background I talked about already. That's the textbook, Ernst Mayr. I'll talk more about him in a few minutes. We're going to do essentially throughout the program a theological anthropology. So let's take it backwards. When it looks complicated, I like to start from the end and work forward. And anthropology is a study of the human person. By the way, a little side on this program, on the entire program, is that added value, you're going to get a little bit of Greek and Latin along the way, right? Because what happens is in the sciences, meaning the empirical sciences, they actually developed about 400 to 500 years ago, and the common language in Europe was Latin. And Latin, in a sense, is a derivative from Greek. So many of our words in the scientific world are have a Latin or Greek origin. All right. So if you have Spanish on the background, it will help a little bit. Anyway, anthropos. Anthropos is a reference to the human. Okay. So anthropoid is a reference to or human-like shape, the primates, including gorillas, orangutans, and chimpanzees, which are our closest biological neighbors. And logos, okay, anthropos. And logi or logos, logos is study of. Originally comes from logos, the word, all right, or grammar. So logos is study, a study of the human. And it's going to be a theological anthropology as opposed to a humanistic secular anthropology, because we have particular beliefs that I hope you will see actually complement and enrich our anthropology. So theological meaning that we're going to have God perspective in mind here, all right? And we're not going to, the one thing that we're not going to question, that I'm not going to question, is the possibility of the existence of God. So if we can all agree on the possibility of the existence of God, then from there we can question everything else that we're going to talk about. So if there is a possibility of the existence of God, there are consequences to that. And one of the consequences is that from our Judeo-Christian perspective, we believe in one God. So it's what we call monotheism, in contrast to many gods, which is known as polytheism. And there's a logic to it, because if one of the characteristics of God is omnipotence and omniscience, you know, that he is all, he, it, she, whatever, this being is all, right, whole, then if he's going to start sharing his power, then he's not fully God, he's a demigod. So monotheism kind of makes sense to a logic of one who is all, whole. So monotheism, which we inherit from the Jews, the great contribution of the Jewish people to humanity for all the duration, is the belief in one God. In contrast with all the other religions that were going on around them and before them, which were tremendous civilizations to the point that thousands of years later, their structures are still here. You know, the Egyptian pyramids, they can be seen from satellite, and the Chinese wall can be seen on a satellite on Earth. You know, the human structures that can actually be seen from space. Chinese dynasties, you name it, they had gunpowder, they had ink, they had silk. I mean, it was incredibly rich civilizations, but they're all polytheistic. The Hindu civilizations, the Roman Empire, even Greece, all tremendous civilizations, and in Mesoamerica here with the Aztecs and Mayas and Inca. And yet, the common factor is that they all believe in many gods. Very rich mythology, but really not too logic, in the sense that these gods had to share power with each other. And so how can they be really God, right? In contrast to all that, the Jewish people, who didn't leave much of a structure, the structure that they tried to preserve was the Jewish temple, which represented, the temple in Jerusalem represented one God precisely. Okay, and that's why it was one. One temple in Jerusalem got destroyed not once, but 2 times. 500 years before the time of Christ, first with Nebuchadnezzar from Babylonia, Mesopotamia, what is today Iraq and Iran, the Fertile Crescent there, went in and destroyed the temple. And then in AD about 72, after Christ, the Romans finally, after several years, also destroyed the temple. And they're working on the reconstruction of the 3rd temple, which is supposed to be the final temple and the second coming of the real Messiah and so forth. That's Jewish theology, all right? But what we do inherit from them is the belief in one God. And then we take it a step further and say, wow, this God loves us so much that he does something that is totally irrational and insane, he becomes incarnate. He becomes a human being like us, and we call him Jesus Christ. All right? So that's why it's a Judeo-Christian anthropology or theological anthropology. All that to say that Christ is our leader, and we believe in him as true God and true man, two natures in one person, and they're totally harmonious and integrated so that he's not schizophrenic. It is actually the human par excellence who we follow to the point of martyrdom. Well, it's in process, okay? And we're all in progress here. Anyway, we pick up from being created in God's image from Genesis, which is pre-Christian. It's actually one of the 5 books of the Torah, the Jewish Torah, and we call it the Pentateuch. You'll get more about this toward the end of the course when I go into the faith dimension and the religious dimension of the course. But we're going to do a Judeo-Christian theological anthropology ontogenetically and phylogenetically. Again, all these words is because in science, we don't want people to know what we're talking about, so we make up these very complicated words. But a big part of this program is to break up in these words and get into them because they are compacted for a lot of meaning, all right? Genesis is origins. So whenever you see genetically, Genesis, genetics is the origin of, and onto is, again, the Greek and the Latin coming in. Ontos is being, the origin of the individual. So the key question here is, when does human life begin, for example, all right, and the consequences of that. So we're going to look at the human individual. When do we begin as human individually, ontogenetically? That is going to be next semester, the course on the beginning of human life. And by association, the end of human life also. So the 2 courses for next semester on the human life, bioethics, and all the issues that are surrounding the beginning of human life, the end of human life. Phylogenetically is a reference to the phylum, which is the group as a species. When do we begin as a species? We cannot deny the fossil record, for example, that the bones are there, all right? And the other lines of evidence for evolution. So I can tell you from the start that in Catholicism, not only do we accept evolution as a theory of process, I'm actually going to teach you some evolution during the semester, all right? And we're going to get into the scientific evidence for the origin of the human species. Okay, so that's the phylogenetic part, meaning, and this is going to involve us basically the rest of the semester, and we're going to complement it with our faith to enrich it and to see how far from science and religion being opposed to each other, they actually complement each other very elegantly and very well. Okay, so the first thing that we derive from being created in God's image, and how so, okay, because one thing that God does not have as God is a body. Yes, the person of Jesus Christ, but God is God. So how do we image God beyond the body that we share with Jesus Christ? It's going to be essentially in 2 great capacities that we have, the intellect and the will. In contrast with the rest of nature, the intellect and the will. So that affords us a particular dignity. And many of the arguments in bioethics are going to be about the dignity of human life, individually and collectively, the dignity of humanity, which is an intrinsic dignity, is not contingent. What I mean by it's not contingent, that it's not based on other people's approval or disapproval or the government granting us dignity. No, we have a dignity that is intrinsic to the human person because we are God's image. Even in spite of ourselves, even when we don't respect ourselves, that dignity comes from God and not from the government or for any particular social group or belonging to any particular society, etc. All right? So this has consequences even at the legal level. And this is where we're going to meet many of the challenges in bioethics, because many people do not agree on what constitutes this dignity of the human person. And at what point in development, right? Should it be from conception, or should it be at birth, or when the teenager gets the keys to the car, you know, or when finally they grow up and they start paying the bills, right? A lot is anchored or posited on when we establish the dignity of the human person to begin or end also in the end of life issues. Okay, so I'm going to try to bring you proof of the intrinsic dignity of the human person and this theological anthropology from the scientific part and from the faith perspective, all right? So on the scientific part, it's going to be essentially delving into two disciplines. It's going to be the discipline of evolution and the discipline of genetics, which again complement each other to explain to us basically how nature happens from one generation to the next. And how is it possible that we have life on Earth? I'm talking about organic life, all right? And on the faith side, well, we'll pick up from scripture and tradition, both. So we as Catholics were not sola scriptura, with all due reverence and respect to our Protestant brothers and sisters. We are not sola scriptura. We are not Bible alone. It's not just the Word of God. We also believe that there is a tradition that word continues to be active in life throughout the 2,000 years of Christian history that we have, all right? And so that there's also a tradition. For example, Jesus Christ never spoke about nuclear warfare. And yet, the Catholic Church today has a very definite position with regards to nuclear warfare. Essentially, we should not go there, all right? So how do we glean that? It's not in the Bible directly. And yet, it is part of our tradition. Number one. Number two, if we take Bible at all, alone, especially fundamentalism, which I'm going to get into, and you'll hear me talk about fundamentalism throughout the course, a little interpretation of the Bible is going to get us into problems, deep problems, all right? Because when we interpret the Bible literally, we're going to find a lot of contradictions in the Bible. Even things that are totally inhumane, like mutilating our body, you know, plucking out our eye or cutting off our hand, which is not what Jesus intended to say. Rather, he intended to say, do not sin. He was using emphatic language. So if we don't interpret the Bible literally, then what is the other option? That we have to use tools of interpretation, what we call the sensus plenior. Don't worry about the words, I'll go over them. I'm just giving you an overall, but the sensus plenior is the fuller meaning of the passage. What did Jesus intend to say, or any scripture passage? What did the author intend to say with what is written there? All right? Including the historical context of the time, because many of the parables and the scenarios, right, what the Germans call the Sitzinleben, the situation in life of the time of the passage, typically they were agricultural societies that were very in tune with nature and not at all technological. So these are pre-scientific societies that had a lot of wisdom, just natural wisdom that came from farming the land and so forth, okay? And they were very familiar with life and death because it was happening in their lives on a daily basis. We are very removed from an environment like that in a highly urbanized and technological society, where when we think of meat, we look at the prime rib that is at the Publix department and not the meat that is off the animal right now as we're killing it and skinning it and so forth, okay? The pork that we grew, the suet that we grew in the backyard. All right, so context is very important for interpreting the Bible. Let's move forward because we have to compress a little bit. We don't really have time to cover the sciences from its origin, especially its old origin back in classical Greece, actually. We're going to pick up on modern science since the Renaissance and what is known as the Copernican Revolution, which I'll cover more next lecture. But now, just a little overview the past couple hundred years, all right, from the 19th and 20th century. As I go along, also because I'm trying to pick up everyone's background. Some are more scientific than others, some are more religious or social than others, just so that we're all on the same page. When we talk about century and the actual day or the actual year or century, there's a numbering convention, all right? So that, for example, then the 20th century is the 1900s, right? It's the 1900s. And the 19th century is the 1800s. So they're equivalent. Don't get confused if I say 1900s is the 20th century. 1800s is the 19th century. So for example, now we're in the 2000s, all right? Beginning the 2000s, we're already in the 21st century. Okay, so 1800s, we have the two big ones, at least for our purpose of this theological anthropology that we're doing phylogenetically, looking at human origins, all right? They're going to be, of course, Mendel and Darwin, Mendel on genetics and Darwin on evolution. And then in the 20th century, so these two fellows, they were contemporaries, but one was in what is today Great Britain, still back then was still Great Britain, and but Mendel was in continental Europe, in Eastern Europe, what is today the Czech Republic, and they were contemporaries, but they didn't really know each other directly, all right? Which is a shame because we could have had what is known as the modern synthesis, the complementarity between genetics and evolution, because basically genetics gives the molecular explanation for the process of evolution to happen, all right? But Darwin didn't know that. So Darwin is phenotypic. Darwin is looking at structures and functions and comes up with a theory of evolution, with a possibility, all right? But didn't know exactly how at the molecular level. Mendel, on the other hand, he's a mathematician and also a monk. I'll talk more about him. He had been able to elucidate and figure out with his P experiments these inheritance factors that he calls these inheritance factors. If they had known of each other and used each other's work, we would have had the modern synthesis in biology, you know, almost a century prior to when it happened, at least half a century prior. They were in the 1880s, and that actual synthesis happened in the 1920s, 1930s. With some people like Fisher and Haldane, which I'll mention, putting together Darwin and Mendel's work, all right? That is known as the modern synthesis, which happened at the beginning of last century. And from there, the explosion of the sciences, not only biology, but biology as a background to medicine and other sciences, ecology and so forth that we have today with the sophistication of satellite monitoring and you name it. Okay, so that modern synthesis is going to open also this new field known as population biology, which is very popular today, all right? Which leads us to ecology when we consider also the physical environment, the physical and chemical factors influencing the population. So this is kind of a little overview. Going a little deeper now, I'm going to look at the 19th century first with Mendel and Darwin, and then at the 20th century. So these are the days. You can see that they're fairly contemporary. By the way, at the end of the lecture, I'll also make a PDF out of, I'll send you the PowerPoint actually. I'll send you the PowerPoint of each lecture, okay? Because part of the program is the idea that you're gonna absorb some of this and integrate it and so forth, and then you'll become the teachers, all right? So you take it on and integrate it to whatever you're doing and pass it on. In fact, the internship, which I'll talk more about the internship next lecture, but the internship basically is engaging other people in bioethical issues. So you can start already after today talking about that. Also starts counting. You need to reach 150 hours of engagement with other people, and that's the internship, all right? So technically that course is gonna appear in the second fall, or you're gonna start on it already from now and accumulating those hours of internship so that by the time you get to the second fall, you've done that, all right? Okay. So Darwin and Mendel, they're contemporaries. Let's look a little bit at their life. Here's Gregor Mendel. He was an Augustinian monk, which was the Augustinian order, the one who founded this university also back in the 60s, all right? Their flagship university is Villanova up in Pennsylvania. And they're not from, so St. Augustine in the 4th and 5th century did not found a religious order, but he did write a lot of books after his conversion. He was a little devil before he became a saint, and even had a child out of wedlock, which he had the audacity to call Adeodato, God-given, on his quest for truth, that he was going through all the Greek academies and all that, secular. Eventually he ended up in Milan, northern Italy, and went into the cathedral. And it happened to be that the bishop of the cathedral was preaching at that time. That was Ambrose, Saint Ambrose, one of the fathers of the church. Okay, and he was impressed. Augustine was impressed, and he said, "Finally, I found the truth." And he kept hearing this voice, Augustine, "Tolle lege, take and read. Take and read the Bible," because the only book he hadn't read at that point was the Bible. And that's where he found the fullness of the truth. He has his conversion. Monica, the mother, after 30 years of praying for the conversion of her son, finally happens. Not only does he become a priest, he becomes a bishop. He's sent back to Northern Africa, where he came from, and then he spent the final 40 years of his life writing books on theology and leading his people. He writes a whole library on the Trinity. Mm-hmm. And so that was Augustine. And even though he never founded a religious order as such, many orders have been inspired by him, including these guys, the Augustinians. So Gregor was an Augustinian monk. He was also a mathematician. His dad was a mathematician. And he has the monastery, of course, the cloister, to play with his peas, right? The plant is a vine. And it turns out that the pea has several characteristics that are what we call all or none characteristics. And he discovered on this pea plant 7 characteristics that are all or none, and that can be seen. They're phenotypic, the phenotype, what can be seen, all right? For example, the peas are either yellow or green, and they're either smooth or wrinkled. And then the leaves, they either stagger, they either are opposite each other or they're staggered. And the flowers, they're either white or light pink. So that's what we call all or none characteristics. Very few characteristics are actually that way. They're called Mendelian characteristics. And it's kind of the simplified version. So the pea is either green or yellow, and there's no blending, there's no in between. So it's easy to count either green peas or yellow peas, and you can get a statistic out of it. You can get a volume, you can get a number out of it. And then you can look at the ratio. And the classical ratio for him was 3-to-1. When he counted peas on a relatively large number of populations, he would get 3-to-1. He would get 75% green peas and 25% yellow peas. Today, we know that we call that dominant versus recessive. And he was intelligent enough to trace it back to the previous generation, so that he figured that the parents were contributing the genetic factors, which he called just inheritance factors, because he did not have the molecular biology that we have today, all right? But he was intelligent enough to figure out, because of the ratios, that the inheritance factors were coming from the parents. In this case, at the cellular level, we talk about egg and sperm, all right? That the egg was carrying some characteristics and the sperm was carrying some characteristics, and depending on whether the characteristic was dominant or recessive, he will get the ratios that he got, all right? So this is showing this little squares where one lines up the eggs over here, the genetic makeup of the egg, actually the genes, the genetic makeup of the egg, and its genetic makeup from the sperm. And of course, it takes one sperm to fertilize one egg, so the combination is what gives us the actual characteristics. Here they're showing 2 characteristics. They're showing color and they're showing the smoothness of the pea itself, all right? So for example, this one is smooth and yellow, this sperm is smooth and yellow, and this egg is smooth and yellow. So when they fertilize, then you get a smooth and yellow pea. But this one, for example, is smooth, and you see the lowercase yellow, that actually means green. It's a recessive, all right, the non-yellow. So when you mix this sperm, which is actually smooth and green, you mix it with this other egg, that is also smooth and also green. In other words, they have the recessive gene on both sides. When they're mixed, you get smooth and green, so the recessive shows. And so these things are called Punnett squares, which was invented by another statistician, Monsieur Punnett. And this is how the characteristics are done. But this is only on 2 characteristics. To show the full gamut, the full range of his study, you would have to place actually 7 characteristics on the egg and 7 characteristics on the sperm, which this Punnett square becomes huge, all right? There's a simplified way of doing that today. But anyway, the bottom line is that you can figure out all the configuration, and then you just count the individuals and you get ratios. Those ratios tell us if the characteristic is a dominant characteristic or a recessive characteristic. All right, the dominant, of course, dominates over the recessive. So here's a photograph of the monks in the Brno monastery where he was living. Brno is a city, a town in what was back then the Kingdom of Bohemia, belonging to the Ottoman Empire in the 19th century, and today is part of the Czech Republic. And before the transition was Czechoslovakia, right? Before the breakup of the Soviet bloc. And here's Gregor with one of his pea plants, probably one of the flowers of the pea. Some of the monks looking at it, others not interested at all. This is an actual photograph of Gregor Mendel, all right, from way back. Here I just listed some characteristics in the human which are Mendelian characteristics, in other words, all or none. And typically they're recessive. So albinism, which is another way for these people who have no pigmentation. So melanin is a dominant characteristic. Melanin is what gives us the color. Then there are degrees of melanin expression. But the lack of melanin is a Mendelian characteristic. Also color blindness, or also known as daltonism, I believe. Daltonism, right? Daltonic. Cystic fibrosis is another Mendelian. Hemophilia, right? Remember the czars in Russia, Tsarina, they were, because of the inbreeding that was going on, they became hemophilic. Sickle cell anemia, which affects blacks in particular. The cell, it's actually, they have a larger resistance to malaria because the trypanosomes, right? The trypanome has to go into the RBC, into the red blood cell, and in the sickle cell has a shape little like a crescent moon or a sickle. There's less volume, less space for the parasite to get in. So, ironically, sickle cell anemia is more resistant to malaria, more prevalent in blacks because it's originally probably a disease that came from Africa. Tay-Sachs, on the other hand, is more prevalent on the Jewish population, I believe, also because of the inbreeding, especially for ultra-Orthodox and Hasidic who all only marry within. But very few are actual Mendelian characteristics. Another one that is innocuous is the widow's peak. Some people have a little tuft of hair in the front, and that is a Mendelian characteristic recessive. Another one is the characteristic to curl your tongue, like that. Most people can do it. Most people, some cannot curl their tongue like that, and that's a Mendelian characteristic. The earlobe, right? If it goes in or not. Most people on the lobe itself down here don't have the little thing that goes in. It's not an actual lobe. It is right. Thank you. Attached or detached, right? Then the dominant is detached, or for some it's attached. These are, you can see that they don't have, in contrast to these other phenotypic characteristics are innocuous. They don't have an evolutionary advantage or disadvantage that we know of, but they're stuck around. Anyway, those are Mendelian characteristics. The important thing about Mendel is that he had discovered these inheritance factors. Okay, so now it can be explained at a genetic level, passing on the inheritance down to the egg and the sperm. Okay, for the next year. To us, it sounds very obvious, but back then it was not that obvious at all, especially when the prevailing thought was creationism, and that God just basically went around with a magic wand creating all the species that exist, including the species that are now extinct because they, like us, they could not deny the bones that had been discovered, or fossil record, all right? On the other hand, we have our friend Darwin. So here is a design, a picture of a drawing of young Darwin in what could be the Galapagos Islands. And here's the Beagle, the HRMS Beagle, His Royal Majesty's ship, Beagle, all right? And the Royal Majesty at the time was none other than Queen Victoria. So that was the Victorian era, which was the great expansionism of the British Empire, including all the colonies down to India and Africa, the Middle East, of course, the Caribbean, etc. To this day, some of the protectorates that they have in the Caribbean. So young Darwin, which seems to be holding a lobster here, and is looking at this frigate bird, a male that has this huge overgrown pouch, which is burdensome for flight, but then it's involved in sexual selection. The female is locked into the pouch of the male, which is somehow supposed to symbolize vigor, all right? But in reality, it's like the tail of a peacock, right? That the peahen is fixated on that tail, and the bigger tail, the more she likes it, but it's so cumbersome for the male that it actually makes the male more vulnerable to being hunted because it's awkward to fly with that thing, right? That's called what's called runaway selection, and we'll look at it later on. But here's John Darwin pondering nature that he sees around him. John Darwin, as he was growing up and had a love for nature, but also a love for God, and many of his teachers were priests. Of course, who were Anglican priests, because this was in Great Britain. And at one point, he was thinking about the vocation, but then his uncle quickly put him into this ship as a naturalist. And the Beagle was going around. The big science back then, in the 19th century, was geology and geography, because they wanted to map the coastlines for trade and commerce. Of course, expansionism of the British Empire and the British Navy and all that. So even for military purposes and for commercial purposes, the coastlines, an accurate map of the coastlines was important. So the mission of the Beagle, which was mostly staffed by geologists and geographers, was to map the coastlines of the world. And that Beagle spent about 5 years going around the world, mostly along the equator, right? You can punch in online and read more about it. But his uncle got him into the Beagle in part to get the idea of clergy away from his mind and get him into science. And so Darwin went around the world in the Beagle collecting specimens from all over the place, and he would collect plants and animals and fungi. Some anecdotes that he had collected all these exotic plants from different parts of the tropical world and put them in boxes, and then when he opened the boxes a few weeks later, he realized that the ants had eaten up all his specimens. He found mushrooms, for example, that were in the Amazon basin, all right? And he noticed that these are drawings from him. Some of these mushrooms were being sold in the marketplace in the streets of London, and these same mushrooms were being used by the Yanomami Indians in the Amazon basin to poison their arrows, the arrowheads. So the same species, depending on where it grew, would either be edible or poisonous. So this is one of the things that got him thinking about, how is it so? Variation is his big thing. So much he goes so big on variation that he actually denies the concept of species and just thinks of variance. For him, he minimizes the issue of species, all right, which has biological implications for inheritance, for the progeny, and he just talks about variance. So to him, life as we see it, again, organic life on Earth is just one big variation from one variant to another, okay? So he took it a little too far, but he did come up with a theory of evolution, which has, actually we need to talk about theories of evolution because there are several, and on some points he's just off the mark, it's not scientific, but he did publish The Origin of Species, of which I'll talk about more in the next lecture. Here's old Darwin, probably what, 60s or 70s at least, but as old as he was, his eyes were still undimmed, and he has that keen vision of seeing variants all over the place. And, you know, when he's looking, how did this frigate bird develop this huge pouch? So selection is going to be big on his scale of value. Okay, so these two guys, there are other anecdotes about them. Like, for example, one of his cousins, Francis Galton, was a physicist in Great Britain. And Mendel eventually published, so Mendel started, what inspired him apparently was a wild, I think it was wild mustard that he saw growing on a windowsill, on the slab of a windowsill in the monastery. Inspired him to think about inheritance. And eventually he zeroed in on the pea plant. It's also said, it is speculated, that way Mendel's numbers are so good, you know, on the percentages. For example, out of 1,000 peas, that famous ratio of 3 to 4, for example, by the numbers, 75%, right? Another thing that we do in this course is we go back and forth between fraction, decimal, and percentage. So it's a little mental exercise. If you feel tired after the lecture, it's a good thing because we'll be doing mental exercise, okay? So for example, just to give a simple example, and I use this actually for the undergraduates to see where they're at with their math. All right, so 1/2, convert that to percentage, 50%, easy enough. Now give me the decimal, 0.5, good, okay. Already you're on the upper 50% of the incoming class, all right? Yes, all right. Then we can bump it up a notch like the cook says, the chef. Okay, so how about 1/3? Okay, well, it's 0.333 progressive, right? But 1/3 is 0.333 progressive, or 33.3333%, right? So there's that extra of the infinite, and so forth. But anyway, so for 1,000 peas, to get the 3 to 1 ratio, right, we need 75/25, right? So 3 to 1 means that a total of 4. We divide 100 into 4, we get 25, so we bump it up to 1,000, is 250. 3 times 250 is 750. So to get 3 to 1 ratio on 1,000 peas, be 750 peas and 250 peas, right? Let's say we're looking at yellow to green. Yellow is the dominant, so 750, the theoretical will be 750 yellow peas and 250 green peas. His numbers were so good that he would get like 748 or 753, you know, versus 251. You know, he was just off by 1 or 2 or 3 peas from the hundreds or the thousands.
Father, on the board,
yep,
where you have, because you're just saying that using those 16, 4 of them are green. The 3 that, because that would be your ratio.
Right.
The darker ones, the 3 darker ones in the bottom quadrant, are
yes,
they considered yellow?
Yes, exactly. Okay. So you see, because you have 2 factors here, 2 independent characteristics, that's another thing, they're independent characteristics. We have to look at it twice. We have to look at that Punnett square twice. We have to look at it for color, and then we have to look at it for texture. Okay. So you come back to it for each characteristic, all right? So let's say just color now. We have yellow, yellow, green, 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 all right? So we got out of 4 times 4, how many squares do we have inside? 16, all right? So we can count yellow. 4, 5, 6, 7, 8, 9, 10, 11, 12 yellow, see? And that's the 75%, all right? So it's gonna be 12 versus 1, 2, 3, 4. So 12 and 4, that's 18, all right? No, 12 and 4 is 16, right? 12 and 4 is 16, thank you.
And then of the
So-
4 green ones, only 1 is texture,
Yes,
which
exactly.
is still
So
the
we,
ratio.
yes, but then we have to look at, so first we look at individual characteristics. In other words, we look at color, 1, we got the 3-to-1 ratio in the color. Then we look at texture, and we should get the same ratio. In other words, smooth, 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 but the wrinkles are here, 4, all right? 4 versus 12. 4 wrinkled versus 12 smooth. So it's the same ratio, 3-to-1 for each characteristic. All right. Now, if you combine them, if you ask the question, green and wrinkle, which is a double recessive, it's going to be 1 out of 16. And now you're asking for 2 characteristics. And so on and so forth. We can increase the argument by characteristics and add a 3rd characteristic, a 4th, the 3rd and the 4th. Another one was short versus tall plants. There are no intermediates on the pea plant. They're either short plants or tall plants, okay? And so you can look at the pea. You can look at, also I think the pod had another characteristic. Then you can look at the leaf arrangement. You can look at the color of the flower, and then you can look at the size. So there's 7 characteristics in total that he found that were all or none. And they're all independent from each other, right? That's what we call independent assortment. Okay. And now, the speculation is that how come he only focuses on this and his numbers are so good and so forth? It is speculated that he may have tried with either other plants or other characteristics of the pea and didn't get the clean ratios. Remember, his background is mathematics. So he's looking for a clean ratio. And if he's going to get like greenish peas that are in between green and yellow, then he discards that characteristic. Or if he cannot tell crisply between smooth and wrinkle, then it's not a clear cut. It's not an all or none characteristic. It's not like a light switch, on or off. It's dim. I cannot tell if it's on or off, okay? So it's speculated that he may have experimented first with other, because what a coincidence, why the pea and why these 7 that are all or none characteristics, okay? Because again, Mendelian genetics is kind of rare. Most characteristics in nature are blended, all right, are polygenic, meaning that there are many genes involved, like height and color of hair, I mean, you name it. It's all ranges, it's all variety. So it's speculated that he had tried with either other plants or with the pea other characteristics and did experiments, recorded the numbers, didn't like the ratios, and discarded them, and never published it, disappeared it. That's a speculation. But it stands to reason. But this one's he published, and because of that, he is literally the father of genetics, because he was also a priest. He was an Augustinian monk, all right? Anyway, there it is. And how did he do the crosses? Well, the flower, is a reproductive organ of the plant. So he would go into the stamen and pick up with a pencil brush, with a paint brush, all right? Pick up the pollen from one plant that had, let's say, white flowers, and he would pollinate. He would cut off the stamen, the pollen of the other flower, so that they don't interfere and don't self-pollinate, and he used the pollen from this white flower into the purple flower over here, and that's how he would do the process. And then wait for this purple flower to grow and put out the seeds and planted those and waited for the growth for a few weeks where he got to pea the new generation. So he was very patient. He would go out there and monitor his plots, right? And the monks will all be fascinated looking at what he's doing, what Gregory is doing with the pea plants today. I'm sure they ate quite a few peas in that monastery, right? As they had all these plants growing. He publishes eventually his ratios on an article in a very obscure mathematical journal, all right, was scribbling all over the place. And Francis Galton, who was a physicist, cousin of Darwin, finds the article and gives it to Darwin, because Galton already knew about Darwin trying to publish, but Darwin was very scared of publishing. He didn't want to publish his theory of evolution because he knew it was going to be a bombshell because of the religious beliefs were going to be destroyed, and he's giving an alternative reason to God creating all the species, all right, just from natural selection coming out from one species to another. And so he doesn't want to publish, and Galton gives him the paper of Mendel, basically to say, look, here is the genetic mathematical evidence for your theory of evolution, a possible mechanism of evolution. The anecdote goes that Darwin discarded the paper because he wouldn't understand the math. Amazing. And basically we're talking about ratios, okay? So I make that story to my undergraduate students to emphasize, get into the math. The more you hate it, the more you have to get into it, because without the math, you're not going anywhere with the degree, all right? Okay, so anyway, we're gonna keep the math as simple as possible, and you're gonna have one of the courses, the only one that I don't teach is the statistics course, the biostatistics, which will come up, is the nemesis every year, right? But here's the silver lining. I do it with you. Every cohort, for the past 3 cohorts, I sit in the classroom and I do the course with you. So I make sure that all my sheep pass the course, all right? If you don't pass, I don't pass. So we do the course together, all right? So take heart, you'll eventually get into it, and we have until the summer to warm up. So I do introduce a few statistical concepts along the way just to get you warming up to that biostatistics course. No problem, we'll do it together. Okay, so let's move forward. Essentially, Darwin's issue is descent with modification. Descent with modification. Think of our parents, all right? We all come from our parents biologically, all right? And we look somewhat like our parents, but not exactly like our parents. We're not clones of our parents, right? And so we have some traits of our mother, some traits of our father, and so we are descendants with modification. Right there, just from one generation to another. Now, fast forward, generation after generation, dozens of generations, hundreds of generations, thousands of generations, all right? That modification, that variation gets wider and wider and wider to the point that today, the people of Africa have maintained, for example, their skin color, the melanin, very dark because they have a large exposure to the sun, which is ultraviolet radiation and so on and so forth. But when you get up to the northern populations, you realize that on the world map, most of the land mass inhabitable is in the northern hemisphere. You get up to Scandinavia, and those guys are as pale as ever, right? They have lost a lot of the melanin because it doesn't have the evolution advantage that it has in the equator, because up on the poles, toward the poles, the incidence of the sun rays has to go through much more atmosphere, so much more UV is filtered, and the UV has much less effect on the skin than at the equator, all right? And so then you get a range all the way from the equator to the North Pole. You get a range, a variation of skin color that matches essentially the exposure to UV over millennia and millennia. And that is also talking about the human evolution, all right? And how one particular characteristic has been affected by the environment. And so we have descent with modification, all right? It's like a summary of what the evolutionary process is about functionally. There's another little phrase that you'll hear me say periodically is think functionally. We're not used to thinking functionally because what we see is not the function, we see the structure, we see structures, all right? But for example, we all know that a door is to open and close. But when we look at a door, just to look at a door, we're looking at a vertical plank that is just sitting there, right? And unless we've experienced the door before, we really don't know that the door is for opening and closing. So think functionally. Okay, so these are the 4 main forces of evolution, or the 4 events that drive evolution, all right? Mutation, migration, drift, and selection. Mutation, which at the molecular level is going to have to do with recombination and mutation and recombination together at the molecular level of the genes of the DNA. Migration, also known as gene flow, which is at the population level. Genetic drift, which just by chance, some species, some individuals within the group, for whatever reason, don't make it to reproductive age, or don't, even if they are at reproductive age, don't get to mate, so they don't pass on their particular genes, all right, that's drift. And then finally, natural selection, which is really the event that happens in nature, is the one that really drives evolution at the natural level. It's natural selection, all right? In other words, we are constantly under selective pressure from the environment to survive or not. And we have become masters of that, mostly because of our technology, By the way, now I see a handout. This is your handout. Like I say, not every time it's going to be as sophisticated as this one, but this is yours for you keep, and you can write all over it. This is your own to keep, so you can follow more or less along with the PowerPoint on the handout. Okay, so the rest of the course, we're going to get deep into these four areas, these four forces of evolution, to understand them well and to see how evolution is possible. All right, and explains a lot of what we call today the modern synthesis. Essentially, biology today is being explained in terms of evolutionary biology, which is not at all incompatible with our faith, as long as we understand both evolution and the faith correctly, all right? All right, so then just jump into the 20th century. Because they didn't really know each other and each other's work in detail, Mendel with his articles on genetics, mostly in mathematical journals, and Darwin with eventually his signature book, The Origin of Species, which is actually by means of natural selection, has a bigger title, known as The Origin of Species, or just Origins. Then that modern synthesis had to wait for decades and decades, almost half a century, until the 1920s and 30s, when mostly Fisher and others rediscovered the works of Darwin and Mendel and made what we call today the modern synthesis, okay? And essentially what happens is that Mendelian genetics, or at least genetics as a whole, gives the mechanism, the molecular mechanism for Darwinian evolution, all right? To explain speciation, the forming and the diversifying of species. That's another process that happens typically in nature. As we see mass extinctions followed by adaptive radiation. In other words, after mass extinction, new niches are opened up, new environments, habitats are opened up, which are then taken up by variants on a species that may eventually lead to a new species. All right, a new species. Also, another thing that we'll be doing at this course, we'll be doing several things at the same time, so we'll be jumping back and forth. One of the back and forth that we'll be doing is going between two models, a deterministic model and a probabilistic model. All right, so we'll be talking in statistical terms on probability, but the fact is that at the end of the day, when we look at ourselves and when we look outside this window, outside this wall, what we see is determinism. In other words, we see determined individuals that live and actually exist live today or existed before, but as determined specific individuals. We're not probabilities, okay? We are specific individuals, so it's forever that tension that exists also in medicine to go, because medicine in principle is a probabilistic, it's a probabilistic science and art. With this treatment, so we have this illness, this pathology, there's been a diagnosis, there's the probability, there's a 90% chance that you will heal, that you will cure with this particular protocol, all right? But it's a probabilistic model. But I may be part of the 90% or I'll be part of the 10%. Until the actual protocol kicks in and we see a result, we don't know where we're going to fall, all right? So forever the tension, and because evolution has already happened, all right, then we're forever into that world of probability trying to apply it to a deterministic world, which is the world of real life that we see around us here and now. And then projected to the future, even bigger unknown. So these are the main actors of the modern synthesis that I've been talking about. Fisher, John Haldane, and Sewall Wright are the three big names that come up. You can look them up online if you want a little further. But also this gives me a segue to introduce Ernst Mayr, which is the guy that I'm really going to focus on which is the author of your book, okay? Ernst Mayr, German. This was happening, yes, mostly in Europe, continental Europe and Great Britain at the beginning of last century. So we're almost 100 years into the modern synthesis, what defines biology and science, the life sciences and the empirical sciences today, with the contribution of the two big ones of the previous century, the 19th century, Mendel and Darwin. Okay, so I want to look at Mayr a little bit more, Ernst Mayr, because it turns out that Ernst Mayr, I'll have a little bit more about his biography next class, but for now, I want to introduce, because I'm still giving you an overview here. Ernst Mayr is the author of this book. So he writes this book around the year 2000, and the man lived from 1904 to 2005, okay? He lived 100 years, and he spanned a very particular 100 years, which is essentially the 20th century. You can imagine at the beginning of the 20th century, even cars were barely around, electricity was just getting harnessed. You know, it's amazing what's happening today. Today we have, talk about cars, we have not one, but two Jeeps on Mars, going around sending photographs, digitalized from Mars to Earth. And here we recompose that wave that gets here and turn it into a colorized photograph, all right? The one rover had been going for so long that the gears wore out on the forward, all right? So from Earth, they send a signal to that rover, to that Jeep, turned the camera around 180 degrees and drove it on reverse. And they got another life out of it. Imagine the technology that we have today, all right? The microscope was barely around 100 years ago. Amazing. Today we have proto microscopes that they can see actual molecules. All right, so the great contribution, so Mayr is a population biologist, dives right into evolution, and writes several books, but this one in particular I love. It's out of print, so what I do is whenever I see it online, I buy it, and I buy these copies, these used copies that cost about anywhere between $5 and $10. It costs more to ship the thing than the book itself, but I keep buying them, and then I have them for you on loan, and I want it back, please, at the end of the course. If you like it so much, you can buy your own copy later. All right, but he's a very good biologist, evolutionarist, population biologist, but I like the book also because he goes into philosophy and theology, and then essentially gives it thumbs down, because as great a biologist as he is, he's a very poor philosopher and a worse theologian, okay? So unfortunately, he comes to the conclusion that religion sadly hampered and slowed down the progress of science. So he comes down on the negative side of religion. Of course, what was happening is a spillover from the 19th century and before, which was basically a fundamentalistic literal interpretation of the Bible. And there was a radical separation between science and religion, and you really couldn't belong to both camps and so forth. So I'm going to be very critical on those chapters of his that have to do with philosophy and theology, because unfortunately, with all due respect, as far as I'm concerned, he got that part wrong. But on the biology and on evolution, I think it's one of the best texts to explain evolution in general. But then when we get into human evolution, I'm going to shift gears into some more recent authors, because they include more recent information, data that we have available. Anyway, Mayr is one of the key points of today's lecture, actually, is responsible for contributing the biological definition of species, okay, which has these two main characteristics, and this is what we call universal characteristics. In other words, all of the species that we've seen so far, that reproduce sexually, they have these two, or actually even the ones that reproduce asexually, like amoebas and so forth. They have the capacity to interbreed. Individuals within a population have the capacity to interbreed and that produce fertile offspring. So that second statement means that one has to wait until the progeny matures sexually and mates to see if they are actually fertile, all right? To give an example, horses can be mated with donkeys. And what do we get out of that? We get the mule, right? Okay, the classical example of a hybrid, but typically the mules are sterile, all right? Or if there have been a few mules that have really been fertile, but their offspring are sterile. So sooner or later, either F1 or F2 or F3 filial generations become sterile on their own, okay? And so that's a dead end. Then the mule technically is not a species, but it's a hybrid. But the horse and the donkey are species because the individuals of those populations can mate and produce fertile offspring, which then in turn produce fertile offspring and fertile offspring and fertile offspring. Okay.
Does it make any difference which parent was the horse or the donkey? Is the male or the female, or it doesn't matter?
I don't know if it does. It's a good question. I don't know. I think that typically the female is the donkey and the male is a horse. I don't know why, but I don't know. No, I wonder if it does make a difference in why one say will produce more fertile than the other.
It makes a difference with the tiger and lion.
Tiger and lion, yeah.
The offspring is totally different.
Yes, depending on male or female, huh?
Yeah.
Interesting.
Even smaller than the two.
Oh, yes?
And the other one is a humongous beast.
Bigger,
You
even.
can see it in the Paragon. It's the one
The zoo?
Paragon.
Oh, Paragon Island, yeah, now it's called
They used
Paragon
to have
Island. Oh, yes?
Yeah, hybrid.
That's called liger,
Liger, they are
the
liger.
mixture between a tiger and a lion.
The other one's
Liger.
called
The tigron? Oh, interesting.
Very
Sure.
small,
Wow.
stout, like a gower.
Yes? Oh, wow. Interesting. Yeah.
If the animal cannot
Interbreed.
interbreed or produce offspring, it's not a species at all.
They're not of the same species. They don't belong to the same species. Okay?
But it's a species, but it's just not.
No, it's not a species. It's a different species. Yeah, it's a different species. Like, for example, tiger, lion, or donkey, horse, right? Donkey is a species. Horse is another species. They're very similar, but they're different species. So
But the mule is not a species.
it's not a species. It's a
But is
hybrid.
that the only, like, the what is it? Is it an animal or an?
It's an animal. It's alive. It eats grass. It does the same thing that the parents do, but it's not a species. I'm talking about the biological definition of species, which is very important because it has significant. We belong to Homo sapiens, to one species, you know, and we cannot interbreed with other species, as close as they may be to us.
So it's dependent on other species to exist. I mean, say a mule isn't going to.
Yes, exactly. It's not going to come up on its own. Exactly. Yes. But the important thing is that going forward, it's a dead end. Yeah, it's a dead end. You know, it's artificial, if you will. It's kind of artificial. It's not. There are a few, yes. There are a few. Most of them are done experimentally. You know, by human interaction. They naturally don't mate. Even populations of the same species may not interact with each other. Different populations of the same species that live in different geographical regions eventually do not recognize each other as the same species.
Like a dog and a wolf.
Well, those are different species. Actually, it's actually a subspecies because the wolf is Canis lupus lupus, and the dog is Canis lupus domesticus. That's a subspecies, okay? But yeah, the dog, and it's also an interesting point because you talked about breed, and the dog is a good example of, it is a species, but it's kind of it's a domesticated species. I don't want to say artificial because they're living, but it's a domesticated species, all right? And you see all the different breeds of dogs, they all belong to the same species. So you can go all the way from a Great Dane, which is like a small horse, you know, to a miniature Chihuahua. And as different as they look, they're the same species. They're Canis lupus domesticus, all right? And if you can get them to mate, they could interbreed. In principle, because they belong to the same species, they could interbreed. You're going to get a mutt. You're going to get something in
But it would
between.
be fertile.
Huh?
But it would be fertile.
No, the breed could be fertile, but it's the same species. You're still within the same species. You're not doing a hybrid. It's a breed. So the difference between a hybrid and a breed. A hybrid is between two different species. They're related, otherwise you're not going to get anything. But a breed is within the same species, just different variants. Different variants. So it's technical, but it's important because it's biological. It has to do with DNA.
It's like a pink rose,
Mhm.
a red rose and a white rose, maybe pink rose, but you'll
Right.
never get a pink rose from pink.
Okay, so pink rose is a breed, all right, but it's also infertile. Okay, there you go. All right, but it's a rose, you see, so it belongs, so it's the same species. It's the same species. So you can get breeds that are infertile.
Right, and you'll have multiple tries to get that particular shade of pink, and when you continue, you might not even get that shade of pink ever again. You'll go more white or more red, or
Fantastic. So it can either be polygenic or epistasis, which is other influences of the genetic.
Thinking of the rose, so
Yep.
a white rose and a red rose, they're in rosa.
Mm-hmm.
Okay, so hybrid teas.
right?
Yeah.
So it'll follow Mendel, so you get 75, 25, and eventually, one will outdo the other, and you'll get the pink, and.
It
that's
starts
why-
degenerating.
And that's why if you want a particular shade of a rose, you're gonna pay Mondo dollars because it's not natural.
It's kind of unique. Okay. It's a special breed, but it's a breed, it's still a breed. So think of this. When you talk about within the same species, variants are going to be breeds, like dogs. How many breeds of dogs? Dozens and dozens of breeds, but it's the same species, the same species. It has the same DNA, the same number of chromosomes, the same type of chromosomes. When we talk about hybrids, we're talking about two species, two different species. They're closely related. You can think of them as, cousins, but not really. I mean, just closely related, but two different species that stand on their own and that they themselves produce fertile offspring, all right? But an intermix of those two species will produce a hybrid. But typically the hybrid will be sterile. So what we're saying is that typically hybrids are sterile. Breeds, interbreeds, may or may not be sterile. But breed is always within the same species. Cannot emphasize that enough. Whereas hybrid is between two different species that are closely related. All right? They may be related at the genus level, they may be related at the family level. We're going there now. So let's go there. This is known then, based on the biological definition of species. This is what is known as the scientific classification. The scientific classification, all right? How we classify animals and plants and fungi and everything that is living on Earth. First, we start with the species itself, which is a double name because the species also contains the genus name. So a little bit it's like first name, last name, all right? For example, I mentioned we're two brothers. So my brother is Claudio and I'm Alfred, but we have a common last name, Chofi, all right? So the last name would be like the genus name, all right? So we have species, then genus. Several different species belong to one genus. Several genera, again, the Latin added value. One genus, several genera. One species, several species, the same. Then several genera make a family. Several families make an order. Several orders make a class. Several classes make a phylum. One phylum, several phyla. And then several phyla make a kingdom. And about 5 or 6 kingdoms make a domain. 3 domains make all of the living things that we have on Earth. So that's the hierarchy of classification, all right, of scientific classification. So we go from small to large or from large to small. What is the most inclusive group of these? Well, I think there are about 7 layers. 1, 2, 3, 4, 5, 6, 7, 8 layers. Which is the most inclusive? The domain is the most inclusive. all living things belong to a particular domain. Okay, there are 3 domains that we'll get to in a minute. All right? And what is the least exclusive, or the least inclusive, or the most exclusive? Is the species, because down at the species level, we're down at the individual species. All right? Several individuals of the species, like for the human, we're about 7 billion right now, but we all belong to the one species. Okay? Okay. It was Carolus Linnaeus, a Swedish fellow, in the 18th century. What hundreds is that? 1700s, right? So 1700s, 18th century. Yeah. Always look at the century we're in, and then subtract 1 from the number. that's how I do it. 20th century minus 1, 1900s. Okay, so Carolus Linnaeus, 1700s, 18th century, and that's another advantage of being online. Is I can just put here. Even if I misspell it, it will correct it for me. And there is Carolus Linnaeus, okay? And he came up with the scientific classification of the species, great contribution. And the idea is that each species has been classified on Earth. We have classified so far about 2 million species on Earth, between plants, animals, fungi, protozoans, protists, unicellular organisms, about 2 million species. That's at the lower end, all right? At the individual end, if you will, of the classification hierarchy, classification scheme. And this is approximately, it is estimated that these 2 million species that we have actually classified and put in formaldehyde in some museum or lab in the world, this is anywhere between 5 to 10% of all the living species that are estimated to live on Earth today. It's estimated between 10 to 20 million species live on Earth today. All right? So where are the others? Well, every now and then, every once in a blue moon, you get the news flash that a new monkey species was discovered in the depths of the Amazon basin or something like that, right? But it is estimated that the vast majority of the species that we haven't discovered yet are, first of all, microscopic, and secondly, most likely live in the deep ocean floor, all right? Several miles below the surface of the sea. And so far, we've only been able to get a few robots down there and beginning to take some samples, and they're realizing that it's very rich, full of life, but mostly microscopic. Many species that have not been discovered yet. So of the 2 million that we have discovered, they have been classified all. So this is a universal taxonomy, if you will, a universal classification that applies, right? Of course, the bear doesn't know it's a bear, all right? And it's an Ursus americanus, that's the common black bear, the American black bear, Ursus americanus, but we classify it that way for the purpose of, because we're fixated, you know, we always want to put things in niches and cubby holes. So here it gives you a kind of a visual breakdown on how the categories expand, starting with this particular individual. We're picking on the American bear today, Ursus americanus. Also, the convention is that, see, the species name includes the genus. What's the genus in Ursus americanus? Ursus, all right, Ursus, which you notice is capitalized, but then americanus is lowercase. So that's the first convention. The second convention is that because it's in Latin and all the names are in Latin, then it's either italicized or underlined, all right? And that's the other convention. Unless we get sloppy, all the scientific names should either be italicized or underlined, and not both. And the genus capitalized, and the actual species end of the name not capitalized, all right? So that's convention. At the genus level, we have other Ursa or Ursi, no, Ursi, I guess it would be Ursus, several bears. Ursus means bear, orso in Italian, oso in Espanol, drop the R, okay? Ursula is a bear woman. Okay, so we have other bears in the United States, for example, or in North America, we got the polar bear, we got the brown bear or grizzly, so it's like a grizzly. So those guys together, they are in the genus Ursus, okay? Go up, put other bears in there like the panda and I don't know what this other, what is it, a marmoset? Or? Spectacle, yes, the spectacle bear, yes, thank you. All right, that's at the family level. So this family of bears includes several genera, which means that some of these are Ursus and some are not Ursus as genus, all right, but they're all Ursidae. Ursidae, all right? Typically when the word ends in DAE or DA, that's the plural, is DAE. But DA would be the singular, that's at the family level. Then, bump it up a notch, more inclusive, to the order, the level of order, we're going to include bears and non-bears. For example, here is what looks like a jaguar, here is the wolf. So these are carnivores. They're about 20, 30 families of carnivores. Anyway, we are part of the carnivores, right? We are in the primate family, primates. Go up from the order into class. We're going to include also other vertebrates that are not carnivores, like marine mammals. These are the cetaceans, like dolphins and whales. And these are the ungulates, the zebras, horses, what else? Donkeys, all those creatures that like to walk on their middle finger, the middle toe. We'll look at that as evidence for evolution later on, the skeleton development. Those are also mammals, right? But not all are carnivores. For example, the zebra is a herbivore, eating grass. Technically the dolphin is a piscivore, eating fish, but I guess could be considered carnivore. Anyway, they're at the class level. We go higher than that, more inclusive yet, we go to the phylum, and the phylum is a significant grouping, if you will, significant grouping, because it's a large group that has some consistency, has some cohesion, all right? And at the level of the phylum, we have the chordates, meaning that we have a spinal cord. Whether it's segmented or not, it's a different story. But at the level of the chordates, we share the phylum with frogs and toads, basically amphibians. We also share the phylum with birds and also reptiles like turtles and snakes and lizards and alligators. And we also share the phylum with fish, whether they're bony fish or cartilaginous fish like the sharks and rays. These are fish. So these are the 5 classes of the phylum. Exactly. Vertebrate is a subclass of the phylum. It's a subphylum, all right? But it's chordate. Technically, the phylum is chordate, meaning a cord, a notochord, a rod on the backbone. Whether it's segmented or not is a different story, but at least at the chordate level, we share it. We mammals share it with the birds, the reptiles, the amphibians, and the fish, okay? All right, we go higher than that still into the kingdom. And now, because these groups are so large, they're fewer in number, all right? So we get to the level of kingdom, and we have essentially 6 kingdoms, animals, plants, fungi, and then the other 2 or 3, depending on how they're classified, are gonna be mostly macroscopic or colonial, like algae, amoebas, protozoans, bacteria, all right? And so, for example, here, this is one of the kingdoms, which is the kingdom of animals. The highest possible layer then is what we call domain, of which there are 3 domains in the world. And inside, if you will, of these 3 domains are all the living species that we have discovered so far in nature, okay? And so here we'll include, in this domain, we'll include other kingdoms like the plant kingdom, as you see here. We'll also include the fungi kingdom, all right? and will also include protists like this little paramecium that is being shown there. So let's take it to one more step here. Here is just the first two categories, domain and kingdom, where we get the 3 and the 6. So the 3 domains are known as archaea, bacteria, and eukarya. Archaea is a reference to archaic, something that is ancient. All these are kind of bacteria, but they're missing a layer that the bacteria have on their cell membrane, and therefore they're considered to be more primitive than the bacteria, even though they look very much like bacteria and behave like bacteria, but they're called archaea. All right, so these are considered the oldest living things on Earth. They go back billions of years. That's 10 to the 9 zeros, okay? Archaea are the most primitive organisms that we have on Earth, all unicellular, but many times they form colonies. So they are in a domain of their own and in a kingdom of their own. Then bacteria, which are supposed to have evolved from the archaea and look very similar to them, but they have this extra layer of polymers on their membrane. So they are classified as bacteria, including what is known as today the cyanobacteria. Imagine my bachelor's in biology from the 1970s in FIU is so archaic than when I studied biology back then, the cyano were the cyanophytes, and they were classified as algae, as blue-green algae, all right, because they were considered to be algae. Think of how primitive I am, and archaic. Now they have realized yeah, that's right. Forget about the fungi. The fungi have to be some kind of plant. We're a plant that doesn't photosynthesize. Into the 90s, huh? Wow, look at that. Amazing.
At that time, they were trying already
to separate. But there's a lot of pushback, right? And resistance. Yeah, the old timers. I tell you, in FIU, when it was just the one building, every 5 stories, I'll tell you more anecdotes about that. They were fun, but the library, went into the library one time going through the stacks, and there was a real actual library with books on the stacks, okay? Forget about computers and anything. It was pre-computer. It was actually one big computer. I'll tell you more about that later. But I go to the library looking through the science, through the biology books. I actually found a biology book that was written in Latin. It was written in Latin, you know, from the early 1900s. Now it's an antique. That would be in the museum. Like the sliding rule. Some of you may remember the sliding rule, right? Yeah, that's in the museum. Oh yeah. See, you guys are all taking. You were born with this thing in your pocket. I know. It's a different world. So bacteria and cyanobacteria, because the other bacteria don't photosynthesize, but the cyanobacteria do, and they actually produce the oxygen that we breathe today, several billion years ago. Together, they can also be classified as the prokarya or prokaryotes. You hear prokaryotes, because they don't have an organized nucleus. They have an accumulation of DNA or RNA, but they don't have an organized nucleus. Whereas when there is an actual organized nucleus with a double membrane, all right, then they're called eukaryotes. Karya or karyon is a reference to the nucleus in Greek. So karyon in Greek is nucleus, and eu is true or real. So true nucleus, real nucleus are the eukaryotes. And they are the protists, the fungi, the plants, and the animals. Okay? So to get a little more sophisticated for a minute, and then I'll back out into simplicity again. Currently, this is the tree of life that is proposed today for the classification, the scientific classification of species, where you have the archaea, the bacteria, and the eukarya. All right? So one little challenge for a moment. See if you can find the animal kingdom. All right, so this is just one layer in from domain. Animal kingdom in here. Give you a hint. They're not archaic, right? Because we're multicellular. They're not bacteria either. because we have true nucleus. So they're going to be in the eukarya. So on the lower left, it's the artist gets it. Right, see? Animals here, Animalia. We belong to the Ophistocontes. Ophistocontes. Very bizarre names. These classifications, and then the plants are actually bigger. Look at plants over here. They are the super kingdom level. All right? But these new classifications are coming out of what we call phylogenetic. This is a phylogenetic tree. You'll become more familiar with it as we go along the program. Phylogenetic tree is based on molecular evidence. Molecular phylogenetics. In other words, degree of complementarity, degree of homogeny, homology. How much homology, how much percentage of the DNA sequence is identical to each other. So to blow you away a little further for a moment, our closest relative of a different species is Pantroglodytes. Pantroglodytes, otherwise known as the chimpanzee. 99% homology. The chimp and us share 99% identity of the DNA, of the 3 billion base pairs that we have in our DNA. 99%. So 1% makes a big difference. Okay? With the gorilla, 98%. Orangutan, 97%. And they're still running around wild with no clothing, climbing trees. And here we are with 2 Jeeps on Mars. So this map is made with percentage homology at the DNA level. Yes, exactly. Because it is estimated, just like these branches, you know, on a tree, we can do 2 things on a tree. We can branch out on a tree all the way out to the leaf level, all right? Or we can go in from the leaf down into the trunk through several branches. First, we go through twigs. Several twigs form a branch, several branches form a trunk, and the trunk will go into a root system that we don't see but has to be there, all right? And so if we go backwards in time, this diversity is pointing toward unity. That is known as the common origin, the common species, individual from which we came. Way, way, way back, about 4.142 billion years ago, because we had to wait about 300 million years for the Earth to cool enough to have a crust on which organic life could exist. And that organic life was going to be an archaea. And so we're going to get into protobionts, the making of a cell, of a functional cell. All right? This exciting stuff, the origin. So this is the big question. There are several labs in the world today that are trying to produce synthetic life from the building blocks of organic material, proteins, lipids, amino acids, carbohydrates, combining them to get a protobiont, a cell that can eventually replicate, make another of its own, and those progeny can make more of their own. Then we have a living organism. Okay, without mention of God, faith, religion here yet. Okay, so how do we get there? Well, we have to go several steps further back, but the first step, the original step, is actually going to be the moment of singularity about 13.5 billion years ago, 13.8 billion years ago, otherwise known as the Big Bang. We'll cover that next lecture. All right? And that's where we allow for the possibility of the existence of God, because from nothing something, ex nihilo, that is definitely a creating act that only a creator can do. Okay. We're going to back to simplify life here, where we're going to look at 5 to 6 kingdoms. All right. So the animal kingdom that we're very much aware of, that we can actually see and feel and smell, etc. The plant kingdom, which is also there. Then the fungi, starts getting a little fuzzy, but when you drive in along the pathway, you'll see there's a nymph ring growing there as you make the final turn into, actually, the next to the final turn, right? Look at the lawn as you go on your way out, opposite the construction. As you make that turn to the left, going towards the chapel on your way out today, look to the left before the turn, and you'll see a set of mushrooms growing up in a circle. It's very rare. We don't know why these things they're called nymph rings. Fairy ring, yeah, because at night the fairies come out and they sit on top and they have their sessions with their wings. Mythology. But we actually have a fairy ring there growing organically, physically, and I've taken photographs of them before. They come out when we have a lot of rain. Okay, so those are the fungi. They're not plants because they don't photosynthesize. They're not animals because they don't really move around. They are a kingdom of their own, and they're called fungi, and they're saprophytes. In other words, they feed on decaying material. Thank God for fungi and bacteria, otherwise we'd be covered with miles of decaying material, over the millions of years that everything has been dying around us. We'd be covered with dead gunk, which, by the way, when those forests die and get buried and they get compressed, they eventually form petroleum, right? Oil. If they compress further, they form coal. And if they compress even further, they form a crystal of carbon that is known as a diamond. We'll get into that later, but that's also all there. Okay, so these are the kingdom. Okay, so these are mostly macroscopic that we can see, but not all. Some are microscopic, okay? And then the bacteria and archaea, which I've added in here. And then this grab bag called the protists, which is a pseudo kingdom today. It doesn't have much, it's not phylogenetically correct, but these are mostly microscopic organisms that have a true nucleus. So they're not prokaryotes, they're eukaryotes. They have a true nucleus. And you have the good old guys like remember the euglena and the paramecium and the amoeba that we looked under the high school microscopes, right? This is probably a diatom from plankton, the algae, colonial or even larger algae, brown algae, red algae, green algae, diatoms, etc. Okay, those are put into a grab bag called the protist, but they're not really phylogenetically related. They're more related by size, meaning microscopic. Okay, so this is kind of a simplified diagram of the scientific classification of the hierarchy of living things. And what we're interested in this course is to look at the origin of these things and how we have these 2 million species going around today, going back on the tree, and the process of evolution itself, including human evolution. All right, now I'm going to give you another hierarchy, if you will, that is not the hierarchy of classification, but the hierarchy of complexity as things are actually living on Earth. That we look at, again, when we look out the window, what do we see? Individuals of a particular species, this is mostly terminology that I'm giving you, okay? Individuals of a particular species, they come together to live in nature, they form a population, okay? So for example, we could look at the population of mockingbirds that live at St. Thomas Forest. Okay, they all belong to the one species, or blue jays. We have families of blue jays in here. So they all belong to the blue jay family, and they live at St. Thomas Forest. So that's a population of blue jays. It would be population of blue jays, population of mockingbirds, population of foxes, population of raccoons, etc., etc. And the same for the plants. We have the population of the slash pine. Several populations of different species coming together, but also populations, different populations of the same species. also coming together, they form communities. So we're getting more and more realistic. In other words, we're getting more and more how it actually happens in nature, looking out the window on the other side of this wall, right? So communities are populations of species coming together in a particular ecosystem, a particular environment, all right? So far, I've been talking about species, right? So I'm talking about living things, whether they're plants, animals, or fungi, okay? So for example, that, what you call that ring, the nymph ring, the fairy ring, yes, fairy ring is here on the lawn. So that's a population of mushrooms that are all from the same species. So that's a population of those mushrooms. Together with the grass that is in the area they're growing and a couple of the slash pine there and a couple of the oak trees that are there, that's a community. Because it has different species, has populations of different species. That's a community. And so far, up until here, we've been talking about the living things. But there are also non-living things, okay? Non-living factors. For example, the air that we're breathing, which is necessary for life, and we kind of take it for granted. But also the ground, the soil that we're walking on, and the temperature, the amount of light that there is. The most obvious change that happens on a daily basis for all living things on Earth is what? Every 24 hours, light and no light, day and night. We kind of take it for granted, but it's a pretty drastic change between light and no light. And it happens on a daily basis. So these are called abiotic factors, non-biotic factors, non-living factors, okay? When we add those to the mix, when we add those factors to a community, we have an ecosystem. So now an ecosystem is getting a little more realistic about life. The particular temperature, the amount of light, the amount of humidity, soil composition, etc. That gives us the ecosystem, all right? So you can see where ecology comes from. It's a study of ecosystems. Logos, study of ecosystems. So we have ecosystems that are in a fairly restricted area, in a fairly, in kind of a local, regional area, we have ecosystems. But then we have larger geographical areas, all right? And when we look at large geographical areas at the continental level, it has many different ecosystems interacting, some more than others. For example, we are a migrating route for the monarch butterflies that come from Canada all the way down to Mexico, through the peninsula into the Caribbean, all right? And eventually they go to the mountains of Mexico where they lay their eggs and so forth. So some regions geographically are very large, and they're part of the niche of the habitat of that species, all right? And so that's a biome. And we have about a dozen biomes in the world, depending on how they're classified, from a tropical rainforest to a deciduous forest to a coniferous forest on the land side. And the same applies on the ocean, because also the ocean, we have the surface, we have the pelagic region, which is just a column of water. Many plants and animals live on the water column. And then we have the benthic region, which is the ocean floor. Too many names, but basically to point out that the biome is a large geographical region of the world, all right? We have about a dozen of them, and we'll look at them more in detail in the environmental bioethics course in the summer. And finally, when we look at the globe itself, when we look at all of the biomes put together, we have the biosphere, which is Earth, planet Earth. So far, the only one that we know for sure there is life. We can speculate about the others, okay? But this one we know there's life so far. And so that's the biosphere. So that is the most realistic that we can get with regards to life on Earth, is to study the biosphere. So because population biology, and we're going to be delving into population biology because the forever question here is, where does selection intervene? Where does selection happen? At the individual level or at the population level? Where actually both are affected. But that's the driver for evolution, right? So yes, it's the individual that's changing, but it's ultimately it's the population that's changing. So two or more individuals of the same species. This is very important. We abbreviate species just SP. When you see SPP is various species, several species, two or more species, okay, SPP. But SP is just species. Okay, Coming up beyond. All right, a couple more minutes, if I may. 3 more slides here. Okay, so biotic factors. Biotic is a reference to biology. It's a reference to life, bios, life. All right, so the living beings, the living organisms, the species that we talked about. Abiotic, A is a negation. Non-biotic factors are the other elements that are non-living, but that affect life and make for the possibility of life. For example, the light, the air, the temperature, etc., etc. You get the point. So we got about biotic factors and non-biotic factors. Together, when we combine these two, what do we get? We get ecosystems, right? Several ecosystems coming together, we get biomes. And all of the biomes of the Earth form the biosphere. Okay, so just basic terminology that we're developing. Finally, what is the evidence from of evolution? What is going to be the evidence that I'll be presenting to you on evolution are these 5 areas. These 5 areas that can be measured. So they're empirical. They can be measured. If they can be measured, then we can say with some degree of certainty that this exists. This clicker exists because I can measure it. All right? That's what is meant by empirical sciences, that they can be measured. The fossil record. Comparative embryology, pointing at common ancestors. We'll compare the embryonic development of various species and see how it relates. For example, at one stage in our embryonic development, in the human embryonic development, around the 8th week of embryonic development, we actually had a little tail bud, all right? That tail bud then is reabsorbed. It becomes what? The coccyx, otherwise known as the tailbone. We don't know we have it until we fall on it. Then we know we have it, big time. Okay? These are fused vertebrae that used to be caudal vertebrae. They used to be in our ancestors, they were a tail, a functional tail. So where did that, why do we have a tail bud in the embryonic stage? Well, because of common ancestry, okay? Phylogenetics, which is getting down to the molecular level of the DNA analysis, comparing percentage homology, percentage of identity with the DNA of different species. We sequence the DNA, we see the actual DNA sequence, and then we match it up with other species, and we see how much percentage homology there is, percentage identity, all right? And that's the phylogenetic tree that I showed you earlier. Also, the fact that the most obvious one actually is the DNA and its variant, the RNA, is the universal code. Whatever species of the 2 million that we have discovered so far, the genetic code, the inheritance code, the package of inheritance is always DNA. This one macromolecule with millions of atoms is universal. How come it's universal? It's pointing to a common ancestor. You know, it started, it worked, it stayed. Okay, and finally, some things like bacterial resistance to antibiotics we're actually seeing with the superbugs in hospitals and other places. It's also an example, it's a living example of evolution going on right now. You know, is the development with superbugs resistant to antibiotics, which is real and a real threat and a real challenge. And so finally, the very last thing is this is kind of an overview. Really, when we look at evolution, we could look at what level, at what scale. Okay, within species, that's actually microevolution. But if we look at populations as a whole and speciation, then we're looking at macroevolution. Macroevolution, developing of entire populations or actually entire phyla, all right, at the large level. So we can look at evolution at the macro level, or we can look at evolution at the large population level. We're mostly going to be concerned with population at the macro level to get at the phylogenetic origin of the human species.
Okay,
Please.
so I have no problem with microevolution because that's, as you said, species can interbreed. But macroevolution, how is that going to happen?
You need two things. You need a lot of time and you need patience. So it's like the old saying, the turtle is slow, but the earth is patient. We're talking millions, if not billions of years, okay? And variation, you also need variation. And then you need selective pressure. And so that gets us into the next lecture. So stick around for the next 3 months, and that's where we're going. To present the evolution, the evidence for macroevolution. Okay, I've said a lot, probably too much. So this is it. I don't know if you have any questions or comments. I will now make a YouTube out of this and email it to you so you can review. Don't forget, by this Wednesday midnight, what do you have? The first summary, okay? In the meantime, also, start reading Meyer. It's the first 3 chapters of Meyer. We're not going to read all of the chapters, but mostly the first half of the book is the most important one for us. So the first 3 chapters, 1 is short with a lot of philosophical and theological mistakes. 2 is a long one, this is 40. And then 3, it gets more into the biology.
Okay, so we're not coming back until the 28th.
Yes, exactly.
Okay, so these are the dates we're actually meeting.
In here, so
And
it's a total of 10
And
sessions.
all of this is going to encompass both courses?
Nope, this is only 513.
Oh, okay.
514 is going to be on top of this, online, on your own. So in addition to this work, you can also go online. I'll send you an email about this one so you have it, okay? But what you're going to do for the 514 is responsible behavior in research and professional conduct, okay? So that one is the one that I'm saying online, even though it's appearing on your schedule, it's appearing for fall 2. But don't wait until fall 2 because then it's going to be compressed. I'm going to send you an email this afternoon with the title of the book and just punch it in online, download it in a PDF, and you can start reading the little chapters. I can pass this around, you can look at it and start summarizing the little chapters. Yes. So you actually have the next 3 months, September, October, November, and half of December to do all of the work for both courses.
Okay,
All right, so they're going both
we're not
parallel.
waiting until this, and then 514 starts. 514 is
Exactly, 14 starts now also. As
That's
soon as
what
you
I
get that
thought.
email, you can download that book and start working. And then in a separate email, I'll send you also the link for the 3 modules of the ethics core at the University of Illinois, all right, the library. That I'll do in a separate. We'll take it by increments because there's a lot to give you at first, you know. Don't want to overwhelm you too much. We'll take it by stages. But I can guarantee you, you'll have plenty of time to do the work.
Father, can you go over again how you want this paper for you?
Right.
If I'm looking at what you gave us as the outline,
Right.
our name, the course,
Right.
the specific lecture title.
Right.
Okay, so I get the margins. Okay, the outline, I am outlining what you taught us or the notes that I took, or
Both.
I'm not sure where you want to go from here. Like I could say, okay, I have a lot of questions. This posed questions for me, which would be my paper to you. Like this opened up, a bunch of things that I'm thinking of.
Right.
But I don't think that's what you're asking.
Well, you can incorporate that, but also give me the substance of what I've talked about. Now, I understand that this first lecture is a little bit of an overview, and I've really given you excess of detail. I don't expect you to tell me the anecdotes about Darwin and Mendel and all that, It's just giving you a lot of detail there. Just the substance. It also gives me a feel for how well you are capturing the substance, because I tend to give too much detail. I tend to give too much.
So I might
Sidetracking.
review by saying we reviewed Darwin and Mendelssohn's, their relationship, or the fact that they did not know each other and they could have, and then mention something about the math, what you call the pure ratio. So to bring these things back to you that we understood what we were getting in the lecture.
So at this point, for example, the great contribution of Darwin was evolution, a theory of evolution, a possible mechanism for the descent with modification. And the contribution of Mendel was, of course, the inheritance factors.
And then we might say something like the genetic drift, which was I gotta find it, my notes, but where you talked about it.
Yes, exactly. By chance, the individual that gets to mate or not mate, and so forth, make it to reproductive age. But
Okay.
even that is almost too much detail for this one yet, because I'm going to be talking about those 4 factors of evolution. The rest of the course is going to be spending a lot of time on those 4. So I don't expect any detail at this point. I'm just giving you, that's the difficulty with the overview, is how much detail to give or not give. Mm-hmm.
One more
Please.
clarification.
Yeah.
The 3 chapters we're gonna read in this is for the next lecture?
Yes.
It's not,
Correct. It's for the next, it's
It's for the
actually
next
for the
one.
next 2 lectures.
okay.
So like chapter 1 for this class, the next class, chapter 2, and chapter 3 for the other class.
They don't match exactly, they don't, but you'll see by topic. It's just that since we have now 3 weeks, we won't meet for another 3 weeks. I wanted to give you, it's the compulsiveness of the professor.
But we could do
Can't
this
stand the
without
students to
reading
have
this.
free time. Gotta give you stuff to do in the meantime. But read up on Meyer, because those first 3 chapters will help you understand the next 2 lectures.
And what was our specific lecture title today?
Today, you can just put introduction for today.
So that's what
Introduction or overview.
and the actual course that we're taking is the fundamental principles in
Yeah,
Phylogenetics. That's
yeah, you
bio with the 513.
Yeah, that's a 513.
Yeah.
So don't give me the full title of the course, not necessary. That's just verbosity, okay? 513 is good. And today's title, Introduction and/or Overview. All right. Again, whatever hand I can give you is yours. You can scribble all over it. Fine.
Concerning that chapter, that you're saying that you think you gave a thumbs down on.
Right, it's there. It's part of chapter 1, and you'll see it. Yeah, you'll see it. You'll see it. Mm-hmm. And but I kind of anticipated there a little bit. I'll give you the reasons why and point out why. Basically, he was living within he was still picking up on a fundamentalistic interpretation of religion. It's a carryover from the 19th century. Mm-hmm. And that's exactly why Darwin was so reticent about publishing his Origin of Species, because he knew that he was going to go against the grain of the standard belief of the time, and he was going to be considered a heretic. And then you see all the cartoons. You've seen the cartoons of Darwin looking like a monkey, like a chimpanzee. And then people misinterpreted him as saying that we descended from the monkey, which is a contradiction in terms, because you cannot have contemporary ancestors. If the monkey exists today and the human exists today, the monkey cannot descend from us and we cannot descend from the monkey, because we're all existing at the same time. It's like saying my brother descended from me or I descended from my brother. No, because we're the same generation. We're contemporaneous. We have common ancestors. We go back. We have common ancestor with a fly, for God's sake. We have common ancestor with trees, because we have living cells. We have nucleated DNA. All right, we get there slowly but surely. We get there. All right, thanks a lot. I apologize that I went over, but any other questions or comments? No, I'm going to close the video now, and this is the other thing that I need to do at the end. Stop recording.